My name is Heidi and I'm a fourth-year student at Pomona College. This video is an introduction to aptamers, what they are, how they work, and why they're useful. Having a good understanding of aptamers is going to help you out a lot this upcoming semester. So let's get started. So, what is this little squiggly line supposed to represent? What is an aptamer? An aptamer is a single-stranded DNA or RNA sequence that is usually about 20 to 100 nucleotides long and has the capacity to bind to a desired target of interest. The target of interest can be a protein, small molecule, cell, or even a virus. In practical application, aptamers can be really useful for detection, among other things. You might be wondering how DNA is capable of doing any sort of detection. As you may know, when DNA has both strands, it tends to form a shape known as a double helix. This is probably the image of DNA that you're most familiar with, because it is how DNA is classically represented in graphics or textbook illustrations. However, it's important to recognize that aptamers are pieces of single-stranded DNA or RNA. When a strand of DNA is not bound to its complementary strand, its perfect match, that is where aptamers have the opportunity to form. Here I have a representation of the single-stranded DNA, which we could also call our unfolded aptamer. These single strands are still made up of the same 4 nucleotide base pairs as double-stranded DNA, A, T, C, and G. However, when there is only one strand, the base pairs are free to interact with each other in new ways, causing the strand to naturally twist up into different shapes. Here, we can see how the strand can match with itself in some places, and not have a match in others. This creates a unique shape and generates what we call the functional form of the aptamer, aka the form of the aptamer that can bind to a target molecule. Here is a visual overview of what we just talked about, where you can see the DNA interacting with itself and then a target. Again, this target can be a protein, small molecule, cell, or virus. Assuming you now know a bit about what aptamers are and how they work, let's talk about how they're being used. In 2021, an aptamer was developed for a specific type of leukemia cell. Now that the aptamer exists, scientists are finding a method to attach a drug to the aptamer, so the aptamer can detect the leukemia and target it better, which reduces the chance of the drug damaging a healthy cell. In this diagram, the drug is delivered to the sick cell, preventing it from further multiplying, while the drug does not get delivered to the healthy one. As demonstrated, aptamers are useful in targeted drug delivery. But how else can we use an aptamer? Moving on to some other applications, here we have aptamer beacons. With aptamer beacons, researchers have developed aptamers with fluorescent tags, so the aptamers fluoresce when they come in contact with the molecule that they are designed to detect. Next, cell imaging. Some aptamers can be used to replace antibodies in current cell imaging technology, and that's primarily being employed in cancer research. Imaging RNA. Again, this is done through combining aptamers with fluorescent tags. Protein detection. If you want to know if a specific protein is present in your sample, you can use an aptamer. Food and water analysis. This involves monitoring contamination, pollution, and the presence of harmful chemicals in our food and water supply. Gene therapy. Aptamers are coming into use with the development of antivirals and vaccines, as they can be used as a tool to measure the effectiveness of a vaccine by measuring the level of antibody production. And then smartphones is here, because there's a lot of work being done right now to integrate technology with aptamers. One example comes from a paper from Wong et al., published in 2022, where they developed a wearable cortisol aptasensor. Cortisol is the hormone directly correlated with stress, so monitoring cortisol levels can be really important for some areas in clinical care. This sensor was an electronic patch that you could put on your skin, and it would use aptamers to detect your level of cortisol in real time. This data would go directly to a smartwatch or a smartphone. Very cool. As you can see, aptamers have a really wide range of uses, because detection and sorting are really powerful abilities in biology, and aptamers give us a way to do so. So why use aptamers? If there is existing technology for detecting molecules, how are aptamers special? I'm going to be unveiling a graphic which was developed based on a graphic published in a paper in 2015, which discusses advantages of aptamers in the biochemical synthesis industry, and it most directly compares aptamers with lab-made antibodies. Aptamers are generally much easier to work with and synthesize than artificial antibodies. They're thermally stable, so they are less likely to denature with temperature variation. There is minimal batch variation, so once you have the sequence for an aptamer, you can accurately generate many more copies. They are relatively cheap to synthesize, and it does not take very long to produce more. In clinical application, aptamers are non-immunogenic. This means that when comparing aptamers to lab-made antibodies, aptamers have a smaller chance of triggering an undesirable immune response, i.e., less negative side effects. They're small, so they travel easily within the body. Finally, aptamers have a broader scope of potential targets than lab-made antibodies do at this point in time. That's all for aptamers. I hope you learned something interesting, and I'll see you in the next video.